Hello. Hello. Good evening, ma'am. Good evening. Ma'am, when will the class will start? Just wait for 5 minutes, then we can initiate the session. Thank you. Let's begin the session. Okay. Okay, let's begin the session. So today we'll study about the targeted gene modification. Okay, I hope I'm audible to everybody. Okay, so in the targeted gene modification, that means we can uh, do the desired changes or manipulation to the genome or to the chromosome of any specific organism, and accordingly, uh, yes, and accordingly we can study our desired findings. Okay, so gene modification means gene disruption, gene knockout. So there are two things: one is knockout, another thing is knock in. Okay, so knockout refers to the deletion of any particular gene. Suppose in your system there is one particular gene which is involved for some particular function. Now you want to study, if you remove that gene from the organism, then what is the consequence. So that particular removal of one particular gene from chromosomal locus or from genomic locus of any organism is referred as knocked out. And another thing is knock in. Knock in means insertion of any other thing to the genomic locus. Okay, so generally knock out is referred to loss of function. Okay, loss of function. And knock in, you can say like gain of function. Okay, like if you suppose you made a mutant of any particular gene, any mutation you did in that gene, and that mutation you want to incorporate to the genome of the DNA organism, and then see what happens when that particular mutation is there, point mutation. So that you do using the knock-in strategy. Okay, so today basically we'll discuss about knockout and knock-in strategy. Okay, so sorry, yeah, tell me. Oh, is it? Hello. Yes. Can you see now? My screen, can you see? No? Can you try logging out and then try to log in? Because I don't know about others. Can others see the slide? Anybody reply? Anybody else see the slide or not? Yes. Okay. Yes, ma'am. Yeah. So can you please try to log out and then try to join the class again? Yeah. Okay. Let's see. Okay. So under targeted gene modification, you study the knockout and knock in of the organisms. Okay, so okay, yeah, so these kind of studies firstly done during the early, or not early, late 90s time. Okay, so for that I think, yeah, So there are three scientists who got the Nobel Prize for the targeted specific gene modification. So let's initiate with some little background. So in 1960, Oliver Smithies. Okay. Okay. So he showed that homologous recombination is can generate allelic variations to the globulin gene, human globulin gene. Okay. So homologous recombination, I hope you remember, we talked about it in the last class. So homologous recombination is nothing but the exchange of genetic information from one chromosome to another or from one sister chromatid to another sister chromatid. Right? I hope you guys remember. So if uh, let's say there are two copies of the chromosome and the genes, these are the particular genes present. So if there is a homology between the sequences, then there are chances of the particular homologous recombination. So suppose at this particular point, because of the homology or the sequence similarity, the homologous recombination is taking place, then it will lead to the changes in the genetic information. So now the gene sequence will look like something like this. Okay, so this can be done via homologous recombination. Okay, so this is how the homologous recombination works. Via the homology, because of the homology between the two sequences of the chromosomal part, there is an exchange of the genetic material from one chromosome to another, and that is how the genetic diversity can be arise. Okay, so this is what Oliver Smith is found for like homologous recombination is one of the cause for allelic variation in the globulin gene. Okay. Yeah. Then after that, in 1977, there was a scientist, Michael Wigler. Hmm. He discovered there is an enzyme which is thymidine kinase. Thymidine kinase can be integrated to the chromosomal copy. Hmm. Hmm. Using the homologous recombination. Okay, this particular gene. And then followed by that, there is another scientist which is Mario Capici. So he further enhanced this particular integration process of one particular gene via the uh, micro injection method, which increased the efficacy of integration of thymidine kinase. Then there is a scientist which is Martin J. Evans. Okay, so he particularly worked in the germ lines. So all others, they also tried the genetic modification, but their work was based on the somatic lines. But Martin J. Evans, he has basically worked in the germ lines, and those germline cells which are pluripotent in nature and can make or targeted for special kind of differentiation further. Okay, so he used this particular germ lines, and you know that when the zygote develops, it undergoes cell division from one cell to two cell then followed by several division, it will go to 8 cell. Then there is a stage of morula, and then there is a stage come where blastocyst is there. Okay, blastocyst. So here in this particular stage, what happens? There is an outer lining, and then there is a division. And there is a division where there is an this after the division, the cell forms this inner cell mass, which are the pluripotent cells. So these are the pluripotent cells which he used as a germline cells. There he introduced these modifications on the genome, and these pluripotent or modified cells he fused into the uh, blastocyst of another mice. And these are called the embryonic stem cells. Embryonic stem cells. Okay. So he was the one who applied this targeted gene modification in the embryonic stem cell, which he further fused. Okay, which he further fused with the blastocyst. The blastocyst of any other mice, and then he was able to introduce or modify the sequences. Okay, and together, yeah, so here it shows first time that these kind of embryonic stem cells can be modified, which followed by, and his work is only followed by the Capici and Smithy, where they introduce the homologous recombination in the embryonic stem cells, and they introduce those modified cells into the mice, and then they can make a mosaic or a chimeric mice for the further studies. Okay, so let's so these scientists, which is Capucci with Smithies, these were the ones who employed homologous recombination in the embryonic stem cells. So, yeah. So he cultured the modified embryonic stem cells with specific DNA in it, or if you'll see the modified DNA where he employed the homologous recombination. Okay, like this was the gene of interest. So using the homologous recombination, he has introduced some mutated copy of the gene which replaced the gene of interest and insert the mutated copy, and these transformed embryonic cell he further injected to the embryonic blastocyst of a fresh mice. Okay, here, and that further get implanted to the mice, which will cause you a chimeric mice. Okay, it will make a chimeric mice where one of the copies of that gene is modified. Okay, Then further, if I talk in detail about this homologous recombination, yeah. So you see, so here you can see that this one is a normal locus where this particular two genes delta and beta are present, and this particular region is homologous to the targeted or the uh, template that we are providing. So what you can do for the modification here is, the first thing here, the plasmid that you select, there should be homology arms for homologous recombination plus a selectable marker. Selectable marker. Okay. So with the help of homology arms, the particular construct will get integrated to the genomic locus, and with the help of selectable marker, this particular construct will also be selected. So for example, here, okay, so there are three cells. Okay. Now what you want, that your plasmid DNA or the template DNA, you want to electroporate or transfer it to the fresh cells. Then there are chances that in some cells the plasmid will not get incorporated. Like here, it did not get incorporated, but it went to these cells. Okay, but this cell is referred as non-transformed cell. In this particular, these two cells are now referred as transformed cells. Transformed cells. Okay, so in order to select or eliminate these non-transformed cells from the transformed cells, you need a selectable marker. Okay, that selectable marker is a drug resistance gene. For example, in case of bacteria, we discussed that there is a antibiotic resistance gene that you introduce in this particular plasmid, and then you provide the antibiotic. Okay, then you provide that antibiotic. So as these two parasites have that selectable marker, they can bear the pressure of the drug and will survive. Okay, these two will survive, but the other cell which do not have any antibiotics resistance, that particular non-transformed cell will not be able to survive. That is how it gets eliminated, and you can select the cells, the only cell which are transformed with your particular construct. Okay, so okay, so suppose there in any particular organism there is a gene, your gene of interest, and this is the remaining sequence. In order to knock out this particular gene, you need to design a construct, something like that will have these homology arms, which are similar to this sequence. In between that, as you want to replace the gene of interest, you introduce something else, like for example a selectable marker, for example a neomycin resistance gene. Okay, that you will introduce in between. Okay, and then this is how your plasmid will be. Okay, so inside the organism you will put this particular plasmid, and with the help of homologous recombination, because of the sequence similarity in these two regions, the replacement of gene of interest with the drug selection marker will take place. Okay, so now so this is your normal locus, this is your targeted construct, and then after the recombination, what will happen, your gene of interest will be replaced by this neomycin resistance gene. Okay? Now your gene of interest is no longer there. and this is you call as modified locus. Okay, this is what is a modified locus. This is how you can knock out one particular gene using homologous recombination. Okay, so this particular process involved double crossover. However, there is also some other techniques where only single crossover can also be done. Like, for example, here is the genomic locus. Let's suppose exon 1. This one is exon 2. This one is exon 3. And then you provide one particular plasmid. And then you have the resistance gene and the other portion of the plasmid. And that's it. Only this one is a homologous arm. Then what will happen? These extra portion of the plasmid and everything will get integrated to the genomic locus using the single crossover. Okay. So this is how homologous recombination may work. And then, so here also the similar thing has been done. So you can see here, it is a single crossover. This is the normal locus. These are the two particular genes. These are the homology arms. A single crossover has taken place, and then you can see after, because the homology arm was after the delta gene, then this will remain intact. You can see this is intact. Then from here, the recombination has taken place. So this portion will come here. Okay. So this has come here, followed by the del beta gene, which is a non-functional gene of this particular one. Okay. Then followed by that, you will have the neomycin resistance gene. Then again, this S sequence, and then the remaining sequence of this particular locus will come. Okay. So that is how this modified locus has been derived. Okay. So this has become a loss of function mutant because there from the beta gene, there is a truncated beta gene has been introduced in this particular organism and the chromosome of this organism. So now the non-functional copy will express, and that will show the effect on the organism survivability. Okay. So it will, yeah. So last class we also discussed the editing by recombinase. So as we studied in homologous recombination, you have to provide specific sequences or homology arms. Similarly, this editing by recombinase is also some similarity with that. So here using recombination, you have this LOXP site. Okay. So Cre is a recombinase which identifies the LOXP site. Okay. So this is what a LOXP site is. So it is a 13 base pair palindromic sequences, okay, which are flanking a 8 base pair spacer sequence. This is a LOXP site which is identified by Cre recombinase. Okay. So Cre is nothing but its full form is causes recombination derived from phase uh, phage 1, bacteriophage 1 virus. So that is why this is a pre-recombinase system which recognizes the LOXP sites. So LOXP sites, the plasmid containing the LOXP sites are given, and then the targeted recombination will take place. So recombination between the LOXP sites, so they will align and remove this particular portion of the exon, and that will cause excision or deletion. Similarly, if the LOXP sequence are in opposite direction, then it will cause the inversion of the gene of interest because of the alignment. Like here in this case, it is aligning like this. Recombination will take place here, and this particular extra portion will be deleted, and this will be the outcome. Similarly, if there is a opposite orientation, the inversion of gene of interest will take place here. And then similarly, if the LOXP site are two different locus, then translocation will also take place. So this is the editing by Cre recombinase system. Okay. Yeah. Okay. So now these LOXP sites are specific sites which are incorporated into the genome with the help of recombination or any kind of plasmid. Then this Cre system is a what you say inducible system. So inducible system means generally the Cre recombinase will not express, but if you add certain drugs or antibiotics, then only this particular inducible, the Cre recombinase will express. Okay. So here you can see there is an inducible Cre system. So in the organism, you put this particular TET-R copy, which normally when expressed is active, and it will bind to this TET promoter, which will express the Cre, and then Cre expression will lead to the modification of the gene. But generally, this is an off system. Okay. So here what you'll do with the help of a drug, you'll keep the Cre recombinase in a off form. Okay, so no Cre expression, then there will be no modification. Okay, in order to activate it, you have to remove the drug. Okay, otherwise it will express. Similarly, this inducible Cre system is also a TET on system. That means in presence of drug, it will active. And here in TET off system, in presence of drug, it is inactive. So in the active form, it will express the actual Cre recombinase, and then it will cause the gene modification. So this is the inducible system. Once you introduce these particular plasmids inside your organism or any particular cell lines, okay, and then you treat those cell lines with certain different kinds of drugs or chemicals, then only expression of Cre will happen, and then only those Cre will perform the gene modification. Okay, so these and suppose if it is doing a knockout, so in expression of Cre or Cre recombinase is doing a knockout, so that kind of system is called a conditional knockout. That means generally this is it is without the induction of your particular recombinase, the locus is not getting modified. But whenever you want, if you provide any certain different kind of condition, then only the knockout is taking place, and that is called a conditional knockout. Okay. Other than that, I also want to say how you select these kind of recombinase or modified locus. There are two type of selection. One is positive selection. Another one is negative selection. Okay, positive and negative selection. So positive selection means presence of any particular gene, okay, helps the cells to be sorted from the others. Okay, so suppose if I'm giving a drug, so let's say this is my gene of interest, Mologia. This is neomycin resistance gene. With the help of crossover, it will be knocked out. Neomycin resistance gene will come in place of my gene of interest at the modified locus. Okay. So there what you will do here, you have selected it by giving neomycin drug. Neomycin drug. So if you will provide neomycin drug, then the cells which have integrated this particular neomycin resistant gene, those only will get selected. Okay, and other cells which have not added this neomycin resistance gene in them, in their genome or in their chromosome, will be dead or will be eliminated. Okay, so this is called positive selection. Presence of neomycin resistance gene is selecting those particular cell population of cells. Okay, so this is positive selection. But then there is also a chance of random integration. Okay, so for example, these are the targeted crossovers. Because of that, these particular things are getting integrated to the chromosome. But also there is a chance that suppose a random break is present here, there is a break present in your genomic locus. So apart from this site, this particular construct will also get integrated here with the non-homologous end joining, where there is no need of any homology. Okay, so at some other locus also, this entire thing, while repairing that by okay, so this thing will also get integrated there. So now this okay, so some cells in some kind of population, these kind of arrangement has happened. In other kind of population, this kind of arrangement has happened, that your construct has got integrated to some other non-specific locus which you are not desiring. So in this kind of cell, your gene of interest will remain active, but these cell will also survive in presence of neomycin. Okay, but only you want only this kind of population where gene of interest has knocked out and neomycin resistance gene has come to that place. Okay, and no gene of interest is present. Okay, so this one and this one both will grow on the neomycin selection pressure. So in order to separate these kind or eliminate these kind of population, there is one more selection which is called negative selection. Okay, so negative selection is what? So negative selection is your gene of interest. Okay. Now this is your homology arm. Neomycin resistance gene, and we'll also have another model kinase. Suppose this one. Okay. Okay. This is model kinase. Okay. So this is a positive selectable marker. This is a negative selectable marker. Sorry. Sorry, I wrote wrong. Sorry, it was wrong. I made. After the homology arm, you'll have this negative selection marker, which is thymidine kinase. Okay, and this is your positive selection marker, neomycin resistance. Okay, so if there is a targeted uh, insertion, then what happens? The crossover will take place between these homology arms, right? So then your locus will be changed to this. Okay, this is a modified locus now. Okay, and this is your target construct. This is your target construct. Okay, now if it is a random integration, so at some another place, what will happen? This cassette will be neomycin resistance and the thymidine kinase. Okay, so yeah, so this is a random integration. So in case of random integration, you can see the entire cassette is being integrated here. Okay, with the thymidine kinase also, thymidine kinase, and then here in the targeted integration, you can see the thymidine kinase is not incorporated here. So now first you employ a positive selection. So in positive selection, you will provide drug, neomycin drug, which will select both this one and this one because both harbors the neomycin resistance gene. Okay, only non-transformant will be separated out. Then you will provide a negative selection. Negative selection. So in case of negative selection, you will give a drug which is ganglivore. So this drug, when given with this thymidine kinase, this phosphodilates this and which will make further toxic intermediates. So the cells which harbors this particular thymidine kinase in their genome, that will make a toxic substance and undergo death. So that cell will be dying, leaving these cells where the thymidine kinase is not incorporated will survive. So this is how these kind of cells will now be eliminated and only these kind of cells will be survive. So that is how you can differentiate between a targeted integration and a random integration. So this is a very important. The positive and negative selection both you can employ and use it for the selection of the true transformants, okay, or the targeted transformants. I hope it's clear. So yeah, similarly, if you want to activate a conditional knockout, so that was a permanent knockout. Via homologous recombination, the gene of interest is replaced and it is knocked out. But there are some genes which you cannot knock out. Without those, the cells will not survive at all. So for that, for some little time, if you want to reduce the expression of that gene, then you induce the Cre-recombinase on the with the help of some treatments, and that only that time that cell will show the phenotype of the knockout of that particular important gene. Okay, so there are several uh, uh, approaches that scientists have classified. So the first one for the targeted modification is a hit and run approach. So here you can, yeah, so before that, there are two types of vector as well. One is an insertion type vector. Insertion type vector. Another is replacement type vector. Okay. So the replacement type of vector we just now studied, it is replacing the gene of interest. So there the two double crossover is happening, that is replacing the particular gene of interest and inserting some another sequence, some point mutation or something else. So that's a replacement kind of vector. Then there is insertion type of vectors. That means, like suppose here in the initial also I have said, between these only the homologous recombination is taking place, and then so this is the genomic locus. This is the genomic locus. This is the vector. So here, if the recombination is taking place, then after the exon 1, here, then this sequence will start arriving. Gene of interest, which you can see here. Gene of interest, then exon 3, then it will go to neomycin resistant, thymidine kinase, and then uh, exon, again exon 1, exon 1, and then it will reach here, exon 2, 3, like this. So this first after the hit, you'll screen by neomycin resistant genes. You provide neomycin and these particular type will be selected. Then after this kind of recombination, because of the presence of two such copies here, exon 3, like this, then also the intrachromosomal recombination will take place. Then you'll screen with some negative selection marker and then exon 1, gene of interest, and exon 3. So here this is eliminated and this is how now your modified locus will look. So this is hit, run. This here the insertion type vector is used. Then there is other technique which is tag and exchange. Tag and exchange, you will use the replacement kind of vector where there are two homology arms. You can see here the homologous recombination is taking place. Screen it by neomycin drug. Then you screen those. Then you provide another replacement kind of vector where you will introduce the point mutation in it, and another crossover will take place. And by the negative selection, because now these with the positive selection you have selected this. Then with the negative selection you will select or eliminate those which will harm, which are these kind, because this thymidine kinase is there. So now these kind of cells will be eliminated and only this kind of chromosomally modified cells will retain. So this is tag and exchange. Then this is a recombination type of vector. So here also, so here the negative selection, they have used this DTA, which I have told that this is outside the homology arm, as I have also explained previously. So here LOXP sites are generated, point mutation is generated, and homologous recombination, with the help of homologous recombination, this particular locus will get integrated here. You can see, you can select it by neomycin resistance and the negative selection, which this one will help in negative selection. And then with the inducible free recombinase, you will able to, with the help of this LOXP site, you will able to remove this particular thing and only put the point mutated gene of interest. Okay, so this is how these three recombination, tag and exchange, and then hit and run system will work. Okay, so let's start with the questions. So this was very simple. In which phase does the replication of the chromosome in mitosis occur? That will be the answer of S phase. S phase is the duplication phase. Now I want you all to interact in the question answer. Okay, so this is the first question. Who created the chimeric mice by injecting embryonic carcinoma cells into blastocyst by reimplanting them into the foster mice? Anybody wants to answer? Yeah, so embryonic carcinoma cells into the blastocyst. So embryonic stem cells. So this concept was discovered by Martin J. Evans, and then with the help of Matthew Kaufman, he fused this embryonic carcinoma cell into the blastocyst and reimplanted them into the foster mice to make the chimeric mice. So correct option will be this one. Okay, Martin J. Evans with Matthew Kaufman was the first to use the homologous recombination in embryonic stem cell culture to target a mutant hypoxanthine phosphoribosyltransferase transferase gene. Okay. So this is a mutant hypoxanthine phosphoribosyltransferase transferase gene, which is a gene which is involved in the salvage pathway. So Smithies, as we have talked, he was the first one to discover the homologous recombination causes the allelic variation in the globulin gene. And after the Evans, Matthew Kaufman and Martin Evans discovery, he employed this HR in the embryonic stem cells and target this mutant hypoxanthine gene to a functional HPRT gene. Okay, so the correct option is, check it. So he was the one. So correct option is B. Okay. So this 38 kilodalton protein can perform efficient recombination at the log-P site in the eukaryotic cells. Okay. We have discussed just now which one will be the recombinase which identifies the log-P system. What is the name of that recombinase? What is the yeah, it will be a Cre recombinase. If it is a FRT site, then it is a FLP recombinase. So FLP recombinase is derived from yeast 2-micron circle plasmid, and Cre recombinase is isolated from the phage, bacteriophage 1, which is also referred P1. Okay, so which of the following statement about the Cre-Lox-P system is correct? Okay, so first one is Cre-Lox-P system consists of two 13 base pair palindromic sequence that flank a central sequence of 8 base pair. Is it correct? Lox-P site we discussed in the beginning. What is a Lox-P site? It is correct. Yeah, it is two 13 base pair palindromic sequence which is separated by a 8 base pair spacer sequence. This is Lox-P site. The full form of Lox-P is locus of crossover, which they are referring as X. Locus of crossover, and P is the phage 1, the bacteriophage. Okay. Second is the Lox-P system consists of two 8 base pair palindromic sequence that flank a central sequence of 13 base pair. It is incorrect. Then production of either excision or inversion is independent of the relative orientation of the two involved Lox-P site. Is it correct? Is it independent of the relative orientation or it is dependent? Dependent. Yeah, so this option is also correct. Last is Cre recombinase is derived from Saccharomyces cerevisiae. Is it correct? No, ma'am. Then it is derived from? Yeah. So this first one is the only correct option. Okay. Which of the following is true for floxed mice development of conditional knockout? So, conditional knockout. Okay. So there are certain terms I would like you to know. So this is referred as transgenic mice. So transgenic mice means that there are some genetic modification has done to that particular mice. So that mice is referred as transgenic mice, or there is no modification has been done, that will be referred as wild type. Okay, wild type. That means no modification. And then if you will do some chromosomal rearrangement, some modification, some insertion of any particular sites, suppose Lox-P sites you have inserted, and now that mice will be referred as transgenic mice. Okay, transgenic mice. Yeah. So here it is just talking about the allelic variation, as we have talked initially, the embryonic stem cells fused with the blastocyst, and then they are introduced to a foster mice, and that will develop into the chimeric mice. So that can be homozygous allele or floxed allele or heterozygous floxed allele. So that is how the phenotype differs. But here it has been talking about the conditional knockout. Okay. So conditional knockout I've already told. Whenever you want to make that knockout, then only you provide certain condition and then only that knockout will happen. That is called conditional knockout. So conditional knockout you can associate with Cre recombinase, right? Because Cre recombinase system, I told you, it is an inducible system. Okay. You provide some LOXP site with the help of homologous recombination. You introduce the LOXP site into the genomic locus. Okay. Okay. So with the homology arms, now you have introduced the LOXP site. Okay. Neomycin resistance. Okay. You have introduced the LOXP site. And like suppose this is the gene of interest, and then you have this LOXP site. Okay. Here and here. This one and this one. Okay. Now you made a transgenic mice. Okay. This gene of interest, now you provide certain condition, express the Cre recombinase, and then only with the help of crossover between the LOXP sites, site-specific recombination will take place, and this entire thing will get eliminated, where the gene of interest will also get eliminated. Now you'll be left with this kind of system. Okay. This is a knockout. Okay. So this is called a conditional knockout. Certain genes which you cannot make a permanent knockout, like suppose if you, this is a very important gene for the survival of that species or a cell line. So if you knock out this particular gene of interest permanently, the cell will not itself survive. Okay. You will not have a live or cell line in hand with the deletion of this gene of interest. Then in those conditions, you provide certain kind of condition, and then only that knockout will take place. And in that, during short duration, you can see what phenotype you are getting. Okay. So this is a conditional knockout. So here you can associate conditional knockout with Cre recombinase. Okay. It is not talking about these floxed allele, it is talking about Cre recombinase. Now you tell me whether it is a transgenic mice expressing Cre recombinase or a wild type mice expressing recombinase. Which one will be the true? Okay, so first you tell me. Here I am made this. So this is a locus. This is a locus. Here no changes has been done in this one. Here you can see LOXP sites are there. So you tell me which one is wild type and which one is transgenic locus or modified locus. This one is wild type locus or not? This one, the first one. Yes. It is wild type because no modification is there. Okay. So this, so the mice which will have this kind of genomic arrangement in that will be called as wild type mice. Okay. And suppose here if I'll express Cre recombinase, whether the knockout will happen? Why? Hmm. Yeah. Exactly. Like not the selectable marker that will come second place, but some specific recombination site, because you are expressing a recombinase in order to make a knockout. Knockout will only happen when you have the sites, those sites for recombination that will remove those gene of interest. So here in the wild types, you can see there is no LOXP site. If the recombinase is expressed, there is no LOXP site to recognize. That's why there will be no elimination of gene of interest. So automatically this answer will be wrong. And here this locus, which is a modified locus. So a mice with a modified locus in its genome, it called as transgenic mice, right? It is a transgenic mice. Now, in this particular one, if you'll express the Cre recombinase, the knockout will take place, right? Yes. Yeah. So that is why for the development of conditional knockout, you need a transgenic mice expressing a Cre recombinase. I hope you understood. Okay, so the vector for knockout mainly contains two stretches of nucleotides with homology to the dash and a selection. What? Homology to? Not restriction site. Here in the option it is given targeted gene. You can say that that is also true. Like as I have all talked to you, that is like beyond the gene of interest I am talking, right? These homology arms I am telling. So these are basically the homology arms which I am talking beyond or prior to the gene of interest. But some people also use the homology arms which is uh, inside the gene of interest. Okay, like this also. Okay, so they provide a plasmid with the homology arm which is in the targeted gene, and then for the selection they have a selectable marker, right? So homology to the targeted gene or the targeted locus also you can say. Okay, so restriction site is not correct. Don't get confused with that. Restriction site is completely different thing. That is digestion and uh, some fragmentation of DNA. That is something else. Targeted locus or targeted gene also you can say, but better word will be targeted locus. Okay, and a selection. A selection. Second one will be a selection marker gene, right? Correct? Yeah. So homology to the targeted locus and a selection marker gene. At least these two things you should have for a knockout. Okay, so which of the following is true about homologous recombination through replacement vector? Okay. So replacement vector we talked. What was replacement vector? Anybody tell? Types of vector we talked. One is insertion type. Another is replacement. Replacement vector. Okay. So the basic difference, if I'll say, in the insertion type vector, you'll see a single crossover. In the replacement type of vector, you'll see a double crossover. Okay. So in case of insertion type, with the help of single crossover, the entire vector portion will get integrated or inserted into the genome of locus. Okay. That is a insertion type vector. And in the replacement vector, you will have double crossover, and the gene of interest or the targeted locus will be replaced with the targeted construct. Okay. So that is the difference. Now we have also talked about positive and negative selection marker. So now I'll read the sentence. You tell me which one is correct. Which one is the true about homologous recombination through replacement vector? So replacement vector, if I'll draw, it will be something like this. Positive selection marker, another homology arm. This is gene of interest. Okay. So this is wild type locus. This is the construct or targeted construct, or you can say replacement vector. where double crossover will take place. Okay. Now the first option, the negative selection marker, which is HSB, thymidine kinase. HSV is nothing but it is a full form of herpes simplex virus, thymidine kinase. So this thymidine kinase is derived from that virus. That is why it is named as HSV. Okay. So the negative selection marker should not recombine into the chromosome. Is it correct? Is it true in case of replacement vector? Yes. Yes. If it will combine, if the negative selection marker is combined into the chromosome, then what will happen? So in these kind of replacement vector, we'll keep the negative selection marker outside the homology arm, right? Because you can differentiate between the targeted integration and the random integration. If the cassette will get integrated randomly at some place, then this negative selection thing will also be there. Okay, so that is why in the true HR mediated replacement vector, the negative selection marker should not recombine into the chromosome. Okay, then automatically this option is wrong. This process will result in generation of mutant that promote gene expression from the targeted locus. No. Then the knockout will happen. So no expression, gene expression from the targeted locus. So first option is the correct. Which of the following is true for an insertion type vector for targeted gene modification? Okay, the homology arm is absent. Is it correct? Is it correct? In the insertion type vector, the homology arm is absent. Is it true or not? Okay. Second is designed with just one arm of homologous sequence. True or not? True? Yes. These insertion type of vectors are performing single crossover. Single crossover means only one homologous arm is provided. Okay? Like here in the previous. Here you can see only this one. Only one is the homologous. Only one arm is provided. That is why after the recombination, this entire thing is integrated and then again back to here. Okay, so only one homologous arm is provided. Okay, so next option, designed with both 5' and 3' homology arms. Is it correct? For the insertion type vector, 5' and 3' means we know like in the DNA sequence, if we'll see. Okay, so the DNA is from 5' to 3', right? The orientation that we see is from 5' to 3'. So the upper portion, this portion we refer as 5', and the lower portion we refer as 3'. Like suppose if I'm seeing only one strand, 5' to 3'. And if this is the location of my gene of interest, then prior to gene of interest, this side is the 5' end. And this side is a this size is a 3' end. Okay, this polarity is based on the DNTP sequences, right? There the 5 prime and carbon of the sucrose sugar is associated with the phosphate group, and the 3 prime hydroxy group is towards the bottom. So this is 5 prime and 3 prime. So prior to the gene sequence is the 5 prime end, after the gene sequence is the 3 prime end. So similarly, prior to the gene sequence, that arm will be referred as 5 prime homology arm, and beyond the gene sequence, that arm will be referred as 3 prime homology arm. Okay, so insertion type vector is having or designed with both 5 prime or 3 prime homology arm, whether it is correct or not. Then this kind of arms are present in which kind of vector? Yes, replacement kind of vector. There double crossover is happening. The homology arm is cleaved by a restriction site. No, it's not correct. So, yes. Okay, so by which of the following approaches subtle mutations can be introduced? Hit and run, tag and exchange, recombination based approach. Actually, all of the approaches can be used for any subtle kind of mutations. Okay, all will lead to the same ultimate outcome, that will be knockout or introduction of a mutation. Okay, all the techniques can be used. Okay, so I'll just let me tell you how we generally make these kind of vectors. Okay, so what you do, suppose there is an organism. Okay, and let's say mice. Okay, in mice you want to delete one particular gene. For example, it is I'll say gene A. Okay, this particular gene I tell you to knock out. So in order to knock out, for this uh, entire discussion, we studied that we need the homology arms, right? Homology arms are needed, and those homology arms you need to put it in a vector. That vector you put or transfect or uh, transform with that particular vector to the mice cell or embryonic cells, and that will develop and give rise to the mice that will be devoid of gene A. Okay, so that is how it is all going to work. So if I'll say knock out this gene, first thing is you have to clone the homology arms. And how do you clone the homology arms? For that, you need to have some DNA that will be having the homology arm. Okay, so nowadays the mouse genome sequence is available online. Okay, the entire genome sequencing is available. There you name any gene, the sequence of that gene will come. Okay, so on the there are database, there you type this particular gene name, it will give you the entire sequence of gene A. Okay, this gene sequence you will get. Okay, so that will be a nucleotide base sequence like A, T, G, C, T, A, many things. It will be several base pairs or something. Okay, from start codon to stop codon, it will give you the gene sequence, the database. Okay, now using this gene sequence, so now what we have studied, that we are making the homology arms before the gene starts, like the in the UTR regions, like the untranslated regions. Okay, the 5 prime UTR and the 3 prime UTR. So you have to retrieve the sequence which is prior to the gene and after the gene. Then you'll perform the retrieval sequence retrieval, that will give you the sequence that is coming before the gene, before the gene starts and after the gene stops. Then you will get that particular sequence before and after the gene. You take that sequence and then like you have taken certain sequence that like before gene A, this portion and this portion, this entire sequence you have now. Okay, now you want this particular homology arms, these two arms. Then what you will do, you have to design primers to make these homology arms and to clone them into our desired vectors. Okay, so with the help of the sequence that you have, now you will make a primers. Okay, so let's see if I can get some sequence. Okay, I'll not be having here, but let's say I'll just tell you, like there is some random sequence. Okay, so this is something sequence you have got. So you'll take to make, if the sequence starts from here, you have to make a primer. So you'll make a primer, a forward primer, and a reverse primer to amplify this particular sequence. You have a template or a genomic DNA isolated from the mice. That from that genomic DNA, you just want this particular sequence, homology sequence, to amplify. For that, you need to have the specific primers that will bind to that locus, and that so that you can amplify only that sequence from the entire genome. Okay, and after you amplify them, you can clone this into the vector. Okay, so you make a primer, a forward primer. Similarly, any sequence, then similarly a reverse primer. So to make a primer, there are certain requirements. It should be at least 18 to 25 base pair long. It should have a melting temperature in the range of 50 to 65 °C. Okay, it should have at least 50% of the GC content. These are all the primers requirements. So between the forward primer and reverse primer melting temperature, there should not be difference of more than 5 °. Okay, so keeping all these things in mind, you design primers. Once you get the primers, you amplify the particular homology arms from the genomic DNA. After you get those homology arms amplified, use them, clone them into the vector. Okay, cloning with the restriction digestion ligation, as we have discussed in the previous classes. And that's how and these vectors are already uh, commercialized prepared vector, where the positive selectable markers, negative selectable markers, and everything, origin of replications are already there. Only thing is, in the multiple cloning site, you have to clone the homology sequence which you have PCR amplified. And you can add some extra restriction site which you can further digest, and that will restriction site will help you to ligate it into the vector. Okay, that is how you can make a homology end clone into a particular vector. And now once the targeted construct is ready, you transfect it and then allow the targeted gene modification. Okay, I hope it's clear. If you want to ask me anything, please ask. Is there any question? Any question, or should I conclude the session? Okay, if there is no question, I'll conclude today's session. Okay, see you next week.